Start. So, welcome everyone. Welcome to today's session. And today we will continue to cover the approaches important for working and understanding the differentially expressed gene. So, before we start talking about the next session, I would like to congratulate the participants who have finished the Introduction to Bioinformatics course. And I'm hoping that everyone here will gradually finish introductory course and transition to Transcriptomics 1. Last time we reviewed Transcriptomics 1 again and started to discuss the output from our RNA-seq pipeline, right? So Transcriptomics 2 builds upon Transcriptomics 1, where we learned about processing of high throughput sequencing data. And in T2, Transcriptomics 2, we will learn about DEGs. So what are DEGs? So DEGs are differentially expressed genes. So genes with a consistent difference between group of samples. That is what we are looking at. So these are differentially expressed genes which are, differentially expressed in different samples. This is very useful because when we compare a condition to a control or compare conditions with multiple replicates per condition. So this was helpful to understand such analysis. So in the last class, we discussed t-test, its history, the nuts and bolts of the formula, and how we can utilize the test on our data. So to review a little bit about the things that we did last time, so we saw that in many cases, especially when we use data generated by someone else, experiments are affected by multiple variables. So factors that we suspect to have an effect on the gene expression. These could be technical factors. In that case, our goal is to dissect the data and identify set of genes that have expression patterns associated with each variable, right? So to understand the principles behind these sort of comparison and how to apply them to our data, we look at our data from a statistical perspective and learn about variance p-values, parametric models, regression, and factor analysis, of course. So here in this example, as you see, a t-test is used as a hypothesis testing tool, which allows testing for an assumption applicable to a population. Okay. So in a t-test, what we see is that this distribution is helping to select the observations that shows differences between two groups with low probability of an error. So what we are looking at is differences, right? So this is the signal that we want to find out, and we would like to, remove the noise, and that noise can be, because the data has been generated by someone else. It could also be due to the fact that there could be different type of limitations within one experiment. So to all the guys who have joined now, we were just reviewing the previous session where we discussed the importance of t-test. So t-test is nothing but a small statistical test which helps us to differentiate between two groups. How can we statistically differentiate between two groups is something that t-test helps us with. So let me just do this. So to understand how these methods work, we did a analysis, and we took another look at the RNA-seq data to understand what observations we have and how to study variations that are observed in a group of samples. So we saw a group of samples last time. We looked at an example where we collected 100 mice and divided into two groups, 50 having this sort of ER positive receptors, and 50 having triple negative triple negative breast cancer subtype. So we injected them into the mice, and we wanted to find out whether a gene expression in the ER group is statistically and significantly different from the TN group. It's just a simple experiment. We wanted to compare two groups and see what sort of, what is happening in there, so that we understand that how these cancer subtypes work. So t-test is something that helps us to do such hypothesis. The t-test produces a p-value that can help us to separate these differentially expressed genes from non-differentially expressed genes. As you see in the slide, this is the formula for the t-test, and we looked into different data sets. We defined two data sets from our breast cancer project, which we discussed earlier, that the data is extracted, how the data is extracted using RNA-seq, and how they have generated data and collected data from different samples from different mouse models. So we are trying to understand here, not only two different groups, but these two different groups have also two different conditions. One is ER positive, another is triple negative. Also, if you have any questions or confusion till now, I'm happy to answer those. Next, we looked into, hypothesis testing. Okay. So we measure the difference between two conditions, and we assume that our null hypothesis to be tested is the difference between the two groups. So we think that there is a difference between the two groups, and that's why we here looking at the null hypothesis, and we are saying that a test of this hypothesis will reveal whether the difference is significant or not. So we are doing this hypothesis testing using t-test. So the level of significance, or p-value, corresponds to the risk indicated by the t-test table. So here the p-value is used often, and represents an unlikely observation to happen by chance. So p-value is something, if it is very high, then it suggests that this event has happened by chance. Since it is not by chance, we can be confident in the signal that observation displays. So if it is happening, within that zone that you see in green color, that suggests that this is not happening by chance. So we do have a kind of a threshold that we decide for p-value. So oftentimes a threshold or a cutoff value used to differentiate statistically significant and non-significant results is usually 0.005. So p-value lower than 0.05 corresponds to less than 5% error, 0.05. So this gives us 95% confidence that our results are statistically significant. So these are all things that we did last time, and today we will look deep into the statistical methods, and in the next class we will start understanding about the machine learning algorithms. So it will be rather simple to understand the machine learning algorithms, but what we are trying to do in today's class is to learn about the data itself and learn about the statistics that involves within that data, the simple statistics or the early statistics, or when you look at data, what you look, how you find differentially expressed genes. And when you have like a list of differentially expressed genes, then you can do further other analysis from a big list, and then coming down to a small important genes using machine learning. But it is equally important to understand transcriptomics 1 and transcriptomics 2, that talks about RNA-seq data in general. So here we are not discussing machine learning, but we are discussing that you should understand what kind of data you are giving to the machine or for the machine learning algorithms. So the important thing is to understand the data itself. So the more you understand about the data, the better your experiments are designed. So thinking about all that, I think today's session will be again talking about the statistics and the data. But it is okay if you, even if you don't understand a lot of it, but you get some ideas that okay, this is how the data looks and these are the statistics involved in it. That would be good start to going towards this sort of learning. So after you go through this session, like you are listening to me right now, when you go to the tutorials and when you do the transcriptomics course, so that you can do within the scope of the program. So when you do that, I think you'll be able to relate more and understand better. So in this session, we will be speaking about, regression and factor analysis. As I was telling you guys that in here, we will be discussing the statistics. So to summarize what all is there in the Transcriptomics 2 course, we have had a discussion and an introduction to Transcriptomics 2 in the last session, and then hypothesis testing and t-test, differentially expressed gene analysis, like we saw in the output from DE-seq. If you remember the pipeline, even if you don't remember, it's there, I mean, in the course. And also we have sent out the file that has the pipeline, so you can review that later as well. Today, we are going to talk about, how to compare these two groups with two different, features, two different factors. So we will be trying to understand that how to find out and understand that two samples that are coming from two different organisms can have different effects on gene expression. So in this class, we will do factor regression analysis and the technical details involved within it, and we will understand how the differentially expressed genes are looked into. So here, I mean, in this figure, you can see a heat map, right? So in this heat map on the left, you can see green indicates increased gene expression. So the green genes numbers have high gene expression, that they have expressed highly in the cell, and the red indicates decreased expression. So the intensity is indicated by the color saturation. So if you see it's like more green, it's high expression. If you see it's more red, it is low expression, very low expression. And that's the intensity gradient. From that you can understand the gene expression in two different conditions. But I mean, if you see this if you see this graph, I mean, if you see this plot, so you will understand that the samples, I mean, the samples that you see here are, single samples coming from one gene in one sample. And this could be random or of little interest because you don't understand much from here. If I plot, if you see this histogram on the okay, so sorry about that. So if you see the histogram here in this image, you will see that each group of the samples and the difference between group means. This represents a difference in expression that you can observe, that two different groups and there is a mean associated with it. So the conclusion of this analysis is, biological interpretation. How can these differentially expressed genes show biological difference between conditions that we study? So this is the hypothesis, or this is the understanding of differentially expressed genes, that in one condition some genes are highly expressed, in condition 2 some genes are down regulated. So how we understand that, what are the important ones for the analysis that we are trying to do? So here you can see, however, a differentially gene expression analysis can be limited when we have multiple conditions studied in a data set. For example, we can compare two different conditions, but we suspect that there are additional factors at play. So, for example, how a disease is associated with age or sex. So, if you are a male or a female, how a disease is connected with that. So that's a factor, right? What is your age? So if you are a 30-year-old guy, if you are a 20-year-old guy, if you are a 15-year-old guy, or if you are a 60-year-old guy, so how can that affect, how can we statistically see the effect of that, the effect of age, how we can statistically see that? Here in this example, we have two different organisms. So we have two different mice models, which we will discuss in this session in much more detail, so that you develop the understanding, and also what we are actually trying to learn here. So we need to learn about these type of relationship, and we can learn about these relationships when we think about the experiment. So let's try to figure it out. I think these lines are still there. Okay, so let's go back to the presentation. So let's just first review the underlying concepts and terminologies we have to understand to study these kind of relationships in our data, right? So we need to understand first about what is a dependent and what is an independent variables. So variables are terms we use to define our data in an experiment, right? So any external controlled variable related to data collection is an independent variable. So it is manipulated by what we intend to study. The data we collect are the dependent variable. These variables represent, one second, sorry about this. So as I was telling you that variables are, this is something very used in statistics very often, and it's just a basic to understand about a few things. I mean, so variables are the terms we use to define our data in an experiment, as I was telling you, and any externally controlled variable related to data collection is called independent variable, and it is, it can be manipulated by our intent of the study. So the data we collect are the dependent variables. These variables represents the measurement we take of the condition in the experiment. So oftentimes we would like to establish a relationship between change in independent variable to establish relationship and measure the dependent variable. So all here what I'm talking about is just to find relationships in data. So it's very difficult to understand when I talk in statistical terms, I think, talking about the variable, the dependent variable, and independent variable. So let's just say we collected data on, for example, eye color. We collected data on gender. We collected data on age, height, and weight of the people. So we collected all this data. Okay. Uh, 1 second, please. Okay, so as I was telling you that we collected data about different things like color of eye, gender, age, height, and weight of people. And we were looking for, we hypothesized that some variables here are related and some of them are not. So how these variables, features from different individuals that we took are kind of related to each other and how they are not. So this is something like we are looking at we're trying to find a relationship. We also think that there are exceptions to the rule. We also know, we think that we also think that there are exceptions to the rule, and how that is. So you know relationships observed in the data can be shown which variables are which are related, which are not related. If related, then how much they are related by. So we are looking at something called a statistical parameter or a number to understand that how they are related. We know their eye color. We have this table. Now we have to find out how they are related statistically. So we are trying to find relationship in data, right? So here, for example, weight and height seems to be logically related. If I plot it, are they in the data set? Weight and height. So you can see they are. So weight and height are how they are related. So if we plot this like you see in this graph, you will find that the trend line is going up. The higher the height, the weight also increases. So if someone is tall, the weight is also correlates with their height. But the dots are not too close to the line. If you can see, some dots seems to be far away from it. So there are exceptions. So some people are thin and long. So those will be the exceptions. So is there a way to quantify this relationship we found? Is there some statistics that can tell us about this correlation and relation, how they are correlated and related? So we collected data and we plotted this graph, and then we looked into correlation. As I was telling you that we are looking into correlation. So indeed, we can find out on how. We can use r square. So r squared is a coefficient of correlation that you can calculate in your Excel as well. So this measures how well the data in your data set is explained by this correlation line drawn through this data. So in this case, we get a R coefficient of 41% or 40%, and 40% is okay, but it's not perfect, right? Our conclusion is that height and weight are correlated, but not perfectly. So let's take another example to see what we learn from this type of an approach. So we are looking at a biological, example here. So this gene, if you see CHREBP, is a gene which is related to carbohydrate metabolism. So in this, if we take multiple genes that are not related to carbohydrate intake, we will not find any correlation between carbohydrate intake and the expression of those genes, because they are not there in the carbohydrate metabolism. Simple, right? However, if we take CHREBP gene which are related to carbohydrate metabolism, uh, we just measure this one gene, if you see here, and the carbohydrate intake, so we can quantify the relationship, right? That by measuring insulin. So if we measure insulin, we can see that there is changes in the expression of the gene. So these two proteins means these two proteins are related, and their levels, expression levels are also correlated, because they are in the same pathway. They are following a mechanism, so one affects the other. That is the takeaway point. These two are related. Now I think you understand this, that how things are correlated, how samples are not correlated, and there is an effect of expression of one gene onto the other, because they are correlated. So we are talking about correlation here. So we can thus study the relationship between insulin and CHREBP gene expression, like we saw in the example. So we can even use this established relationship between the two of the study, additional variables. So for example, if you might know that when you do a blood test for diabetes, you take samples from one condition where you have taken the food, you have where you ate and then did the you know blood sugar test, and there could be a condition where you have been fasting, and then you deposit when then you did the test. So there could be different conditions also associated with this gene expression. So one condition could be where you ate, and one condition is when you fasted, when you did the fasting. So some shows positive correlation between the gene expression, some could show negative correlation, and some could show no correlation. So this is an easy example for you to understand the concepts of two different features and two different genes. So we talked about here two different genes, we saw different states. Now how can we classify them, or how can we statistically analyze them using regression? So there could be different type of relationships within that data, right? You saw that there's a positive relationship where, a positive relationship is that as the weight is increasing, the height is also increasing, or when the height is increasing, the weight is also increasing. And negative, similarly, you saw negative relationship and linear relationship. So such relationships can be of several types. So negative, positive, non-linear. So linear regression is a way to further model such a relationship to determine a mathematical function, and that explains the relationship between the dependent and the independent variable in the data set. So if there is a relationship such as function is such a function is predictive. So based on that relationship, I can predict stuff, right? If your height is this much, I can at least assume that your weight would be around this much. That is the prediction I did based on the data, right? So if there is a relationship, such a function is predictive and will inform us how the independent variable will change if we change the dependent variable, right? If I change the height, how the weight would change. Simple as that. So the goal of regression analysis is to find the right coefficient for this formula that minimizes prediction errors, and also that is completely based on the data. So what we are trying to do is to predict things, and based on the correlation that we see in the data. I think this is clear so far. If you have any questions, please write in the chat, and also you can ask me here directly. So as I was telling you that what we are trying to get to is that how these are correlated, how we use regression to understand how we can predict using the data that we have and finding the relationship within that data. So just simply now we will look into the regression formula, and this is, for example, is your dependent variable. This is a constant, this is the coefficient, and this is your independent variable. So this is the mathematical formula. The regression formula has both the dependent and independent variable in a mathematical equation, plus a constant, intercept, and a coefficient. So here in this example, you see one R square value is very less, where you can compare the height versus age. And another example is weight versus height. So you see one example gives a correlation 0.0238. So the regression line is the line that minimizes the sum of squared vertical distances. So if you plot it, and if you do this sort of statistical analysis, you'll find out that the regression line is trying to find uh, it's trying to find a line that correlates well with your data, and that from which it can derive a coefficient that explains your data correlationship, the relationship within that data. So we discussed here factors, right? One factor age, one factor is height, one factor is we explained through insulin example. Also we saw that fasting and non-fasting state. So factor analysis is an adaptation of ANOVA. So ANOVA is a statistical test, which made more accessible by configuring the variables according to experimental factors. So this is what factor regression analysis is all about. And as you saw that in data, we have not just one type of data, within that data, there are different, types of data. So we have different factors. We call that additional factors. So it's not just, continuous data, but it is also categorical. For example, fasting, eye color, those are like differentiate the data into different levels because they have different conditions associated with it. So you have to understand that you have to differentiate these because they are coming from a different condition, right? To study the relationship between these variables and the data, and to understand that what could be, how can we talk statistically about this kind of data. We do factor regression analysis, and we convert this categorical variables into numbers that can, define, our analysis. So now we will go into our example, which we started working on for a while, about the, types of breast cancers. So in our example, we use the same data set that was used for differential, expression. The breast cancer samples were obtained from PDX models. So these PDX models are, immunocompromised mice with breast cancer xenografts. So breast, different type of breast cancers, were implanted into these mice. So a total of, 4 different mouse models used in the original study. So the 4 types of mice models are SCID nod. So mice, SCID nod mice have severe, combined immunodeficiency. So they don't have uh, T cells, they don't have B cells. So SCID CB17 is a mice that have, also, severe combined immunodeficiency, but they have natural killer cells, macrophages and granulocytes. Arithmetic nude mice lacks a thymus and are unable to produce T cells. And XID mice lacks a thymus, but it resulting in no functional T cells, but they have, mutant B lymphocytes. So these, all these, different mouse models have different, characteristics. They are used, they are, prepared, they because they help in understanding and learning about our body better, because we are trying to mimic different conditions here, right? So this is the data set that we looked into, and this is the data set which we started to work on, and we wanted to, do the analysis on it. So going back to the data set, we in our study we looked into, ER positive and triple negative. So we looking into 2 mouse types and 2 cancer types, right? We looked into ER positive and triple negative. So since the type of mouse affects the microenvironment of the tumor, mouse type could also impact our results. This is what we learned so far, right? That different factors, different conditions. So here, 2 different type of mouse. So the microenvironment of the tumor would be different, and that could impact our results because we are looking at different models. So the different mouse models, such as SCID versus arithmetic nude, might affect the gene expression of the tumor, right? Therefore, we will perform a factor analysis with, breast cancer subtypes as a factor and PDX mouse model. So one is breast cancer is a subtype is one factor, because you can see that there are 2 factors. One is ER positive, another is triple negative. Another factor is the mouse PDX models, which is SCID versus arithmetic nude. So we will name them like this, and we will understand that there are 2 types of factors. Now, next step is to do this sort of analysis. So what we are trying to look here is that what could be the correlation within this data. So Richishmita, are you having problems? I think you're constantly logging out and logging in. Is there any issue with the internet connection?
Yes, sir, I'm having problem with the net connection.
Okay, so I think we will also do the recording, so we'll send that out. So I'm sorry about that problem that you're facing. Anyways, Soham, do you have any questions so far? We will go into the pipeline now.
No, no, I have no question.
Thank you. So, what I was telling you that first you need to prepare the pipeline, and it is very simple. Just we have to go to. I will do the demonstration right now, and also I think I have emailed you the links. Let me email you the links again. The file. This file is nothing but the file that we used last time. That we Thank you, Soham, for this sharing this paper. So this is the download link. That I will just share it. This is the download link for the data. And also, like, if you want to do the t-test yourself in Microsoft Excel, I think you will learn more about it. So this is the link to do the t-test. And this is the link to the, factor regression analysis that we talked about just now. So now we are going to the server. So this is the link to the server as well. Okay, so all the links are here. Perfect. Now let's look at this. So let's just first figure out from this image, and then we will go to the slide. Which is the link for the file? Which one is the link for the file? The first one, the tiny URL. I'm going to paste it again then. Sayok. This is the link to the file. It's a text file that you will download from this URL. So you can save it as a text file, or even copy paste it in a text editor, whatever. I mean, you can just download it and it will be saved as a text file. So the name of the file is so the name of the file is, wait, is this one? No, this one. So this is the name of the file that you will get after you download the file. Okay. So now going back to the session. So to prepare the groups, okay, now let's go to our platform and do the analysis. So this is the platform. Hi, Sarah. Thank you for joining. You're a little late though. And we are doing factor regression analysis together to give you an update. So in here, there is this factor analysis. So if I go back, there are different areas of analysis, if you can see, and under the data mining section, there is called factor analysis. So you click on the factor analysis, and then you click on start. You don't have to upload anything. You just start clicking start analysis by clicking on the start button. Sayok, I wasn't able to download the file. Okay. So what we can do is that we can email it to you, Sayok, and then you can try it again. So we will email the text file. Soham, can you download it?
Yes, I have downloaded it.
Can you send it to Sayok?
Yes, so Shayak, if you can share your email ID, so I will share the file.
Sayok, if you can share your email address, then Soham can email you the file. Okay, so how do we do the analysis? By this sort of comparative analysis. So Soham, Shayok has shared his email address. So if you could please send him the file
Yeah,
also.
yes, just in 1 minute.
Sure, sure. No, take your time.
Uh,
So what I'm showing you here is that we did the start button. So let me go back again. So here, factor analysis, we click on it. We did the start button, nothing else so far. And we clicked on factor analysis. So here also you see that there are not much options. So it tells about the things that we discussed already. You just put save here, and then you here please add the factors and the number of levels in each factors. So you saw in this presentation here we have ET arithmetic and nod mice, and we have triple negative and ER positive. So we have what? We have two factors and two conditions, right? So two factors and level in each factor. So two factors and two conditions. So here in the pipeline, we will be trying to do the same thing. So here we have two, here we have two. Now next, now it is asking to upload the file. Now we can add file. So this is your file. It's getting uploaded. It's uploaded already. So now it will check. Thank you, Soham, for emailing. Now here there are 8, files that are there in the one. So if you, hover your mouse around it, you will see ER node. So put it in here, ER node, put it in one place, ER ATH. Then with the same name, we put it in the same group. Simple as that. Triple negative node mouse, triple negative node, and then triple negative ethnic, triple negative here. So we have put it like different factors: A1 B1, A1 B2, A2 B2, A2 B2. Okay. So let's check what is there in the presentation. So we have just, that's okay. I think it's based on the data. It does not matter which is where. So we can push continue and then end. And then we can save the pipeline by keeping a name called factor regression analysis and mouse PDX and 2 factors. This much is enough. Save. Now we can run the pipeline. And once your pipeline has started, it will show this like this, that it has started. And now you can look at the pipeline by going into my pipelines. So it's running, right? So first, what we did is we need to, yes, Shayok, I can repeat it again so that you can also repeat it. So you downloaded the file, right? Now you go to the server using this link, which is shown here, which I shared. You go to the server and then you click on the factor regression factor analysis. So once you click on the factor analysis, this will come up. Then you click on start and then you click OK. Then you click on factor analysis, then you save. Now it will ask you to add the factors which we discussed, because we have like two types of mouse and two types of cancer. So we have 4 things here, right? Thank you, Soham. This is very informative. And then now it is asking for the file to upload. Now I will use this file which I have downloaded, and usually you will see the groups here, but I think something with the download, it's not it's showing undefined. But when you hover around it, you will see the name of the file. If you can see the name, I can see the name of the file here. So arithmetic nude and node. So the nude mice, what I will do is put them into different groups. So ER node in one place, ER ATH in the other one. So both the ER are here from two different mouse, same cancer. These are two different mouse with same cancer. Triple negative, ER triple negative. Okay, two different mouse. ER, ER, different mouse. Okay, continue, and then end, and then you can run your pipeline. Save, then you save a name. So Shayok pipeline, and then I can name it like factor regression analysis, first try, learning, anything. Save and then run. When I push run, it started to do the analysis. And now I see that this pipeline is processing, so it will take a while, but I have done it before. So I also have that file to download because the analysis is done. So once the analysis is done, when it is 100%, you will get a email that so once the pipeline is done, you will get an email about that pipeline has finished, and you can download this file, which is the output file, right? Now, if I open this file, so when the pipeline is finished, you get a file like this. So let's just open the file. So initially the file that we did start with is this file, which we uploaded. So here there are 8 samples, right? Which we distributed amongst, 4 different conditions, 2 in each. This 2, these 2, these 2, and these 2. We did that, right? This is the file that we uploaded. And now let's look at the file that we had 8 columns here, 8 columns. Now the file that we downloaded, let's look at that file. This is the file that we downloaded. So let us just open it with Excel and see what is in there. Okay, now we have more columns. 1, 2, 3, 4, 5, 6, 7, 8. 8 we had already. 1, 2, 3, 4, 5, 6. Now we have more 6. So 6 more columns. So when the pipeline is finished, we will get a table output. Once you open a new table output from this factor analysis, you should notice that you have additional 6 columns present in your table. So these includes, t-test for each of your factors, factor A, factor B, and combination of A and B. So you have, t-test for all of these done using the factor analysis algorithm. And then you will find f-test values for the same factors. So f-test is nothing. F-test is similar to, t-test, except we are comparing sample variances rather than the means. In the previous one, in t-test, we were looking at the means, and here we are looking at the variance. So in addition, f-test can be used to test the differences between multiple groups. So f-test columns gives us an output of a p-value, which can be used to find statistically significant genes with variation between factor levels. So that is what the output is. So the output is pretty important. That is what we are trying to get, is that which ones are important, which ones are not. So you have different tests telling more about, to tell you more about it. So if we go back to the presentation, we did that, we run the analysis, and then we got an output like this. Here, if you can see, just a second. I think Ritesh Mehta is again trying to join this issue. So you see there are 3 columns that talks about the f-test, right? So let me admit her 1 second. Okay. So in this one, you see, while each of these factors are going to, may have 2 levels or groups. So here, if you have 3 levels, you would have three t-tests. like A1 versus A2, A1 versus A3, and A2 versus A3. So likewise, this is the result from your F test. And now what you can do is that you can look into this data and start plotting to understand what is in there. So the next thing to do after this is to understand how they are biologically important, and what kind of information you can learn from them using this sort of statistics, and coming from these genes. So the genes, you can see the genes, their ID on the left side, extreme left side. You can see the genes, you can see their expression levels, and now you can also see the result of F test and T test in a same place, in a single place. Now all you have to do, all you would have to do is to plot these numbers and see what they mean. So if I do that, if we filter using this group, we can select those genes that are only influenced by factor 1 or factor 2, or between like both the factors. So you can see here. Okay. So you can see here that interaction between the factors is also there. So how two factors are influencing each other. So that is the third column. Okay. So here in this example, gene that is only affected by factor 1, cancer type. So our first factor was cancer type. Second factor was the mouse model, which is the mouse model. So first factor, we looked into the cancer type, and then after looking into the this after looking into the graph, we and plotting those numbers, we found out that a couple of genes that we selected shows upregulation in triple negative cancer, no matter which mouse model we use, by the way. So this is the factor regression analysis. So it is eliminating the mouse factor, and it is just giving you information about one factor, which is the cancer, right? That is the information you got using this factor regression analysis. So so as I was telling you, this gene PGPE-P1L, okay, is a gene that encodes a protein which cleaves amino terminal pyroglutamate residue from protein substrates, including it has a complex mechanism. So on the other hand, you see FOXA1, that is a gene which is upregulated in ER positive samples, regardless of which mouse model. So if you learn more about these genes and their contribution in pathways, you will understand that in one cancer why they have this effect. Then you will understand that this gene might be important for the cancer, might be important for maybe a drug target. So all those things that requires a lot of research, by the way. I know that many of you are here like going into and learning about this thing for the very first time, and many of you are like starting to do this biological learning, first year in BTech, first year, or maybe second year, or maybe final year. But the thing that is important here is that what we are trying to tell you is, this is how research is. Okay, this is what you will learn in coming days, in your future. So it is better so that you understand it, have some understanding about it from right now. And trust me, that will help you so much in your future, because you will be one step ahead. So that is the also the purpose of this program, to make you guys more uh, uh, you know uh, scalable and ahead of many people who, because you have taken this program and you are going through this research experience, which might be difficult, which might be like many people might be thinking that I don't know all this. So yeah, that is all right. We all learn through experience, we all learn through practice. So the point that I was telling you, that after the analysis, after we saw that table and did some plotting, we found out some interesting genes that could be critical for breast cancer. So you can do it yourself, like a good quality research using the available data set. So that is like an opportunity. Okay. So this is one way of doing biological interpretation, by knowing that how things influence. So we saw that in one condition, ER positive, the expression of the gene is very low, and in triple negative, the expression of the gene is very high. And in another condition, we found another gene in which the expression level in ER positive is very high and triple negative is very low. So this makes it important, um, important finding of our result, which we found out after doing factor regression analysis, and by plotting those information that we got from that Excel sheet. We talked about two different factors. We talked about one factor where there is no influence of mouse, and we are looking into only cancer. And then we can look into the mouse and see that factor B, where we will find genes that are differentially differentially expressed in two different types. So for example, here, not think about the mouse, but think about male female. So there's a male, there's a female, and they have different sort of expression levels. That could be a research question, right? That could be answered. So factor 2 is equally important. When we are looking at factor 2, we are looking at what difference is there in the factor 2 data. So two different mouse models have two different influences of gene expression onto them. So how to do this, how to learn more about the genes that you found out. You can do David annotation. So David is a database, right? And it gives you annotation based on the gene ontology. So when you push these numbers in David, when you put these numbers like these numbers, the gene ID, it will tell you what kind of genes they are, what is their function, and stuff like that. So David is also described in transcriptomics 1. So once you find these genes, important genes, you can use David to learn more about these genes. Yeah, thank you, Soham. And I think you can also tell them about David, I think, which is a very useful resource to annotate these sort of samples. So coming down now, let's, like, I think we have like had a long session, and I don't want you to bore you guys more. But to conclude the lesson, in this lesson we learned about a dependent and independent variable. We learned about correlation and regression, and we did factor regression analysis and PDX model example. So we did a lot of things, and I'm really proud of you guys to be you are here and trying to understand what is this whole statistics and machine learning and all those things would be. And it would be really exciting when you start learning about them slowly and ask us questions. So it would be like really exciting for us also to learn with you, learn more, and maybe brush up our one of the basics. Many times we forget to brush up our basics, and we forget many things. So while answering many of your questions, which might come from a very basic level, it helps us also to learn more about it, learn more in depth. So please ask questions, and let us know if you face any problem in completing the courses or understanding any of those things. So I think the Q&A session, we've been trying to ask you, but you are not asking so many questions. But that is also all right. I mean, you can always write those questions out, tell us, email us, put it in the group, put it in the forum where you are seeing your progress. So there are multiple ways. So it's up to you. I mean, it's totally an independent thing. Let us know if you face any problems, and we will be here to help you out. Even when the course is done, even when it is over, you can always write to us, and we will be able to help you all the time. So in the next class, we will be doing machine learning, and we will be talking about how the data is next used for machine learning, and where how this data is applicable to machine learning. And we will also learn about another data set. We did this mouse PDX model. Now we will do another one, which is modeling precision, precision medicine and modeling project. That would be also exciting. So uh, I think once we review all this, and you are able to finish some of the pipelines, some of the course, you will learn in a faster pace. And thank you for joining today's session. And I hope to see you on Thursday, and where we will discuss about data mining and machine learning, and how machine learning is changing, this whole data driven world, and also the biomedical world. I mean, recently there was one drug that came out of this analysis in 12 months. So usually it takes 15 years, and now we are cutting short. And the translational part will come in coming days, and you'll see more and more, and you'll see so much news about all this. So it will be better and best if you learn this and have some basic understanding, so that in the future you can make the most out of it. And thank you again for joining today's session. And have a good night, and we'll see you soon. Thank you, Soham, for putting the David link. And thank you, everyone.